So I just want to thank everyone for coming tonight to the Transplant Journey Inc. And I'm really appreciative of all you guys taking your time out on your Thursday evening to join us all with Dr. Marcus Pereira, Dr. David Ho, and Dr. Anthony Bowen for taking part in this. We're going to be discussing the long-term effects of COVID, bird flu, and the immune compromised population and its effects on us. We've all been, I'm sure, affected in this in some form or fashion. Obviously, now with bird flu coming into the forefront of all of this, we all have some fears and how we've been affected by this and how moving forward it can greatly impact us as being the immune compromised population. So thank you so much for coming to our aid. Dr. Pereira has been ever present, coming to deal with all of us for the past 4+ years. He's been really impactful for all of this. Dr. David Ho, thank you for coming to our aid also in this, and Dr. Pereira for arranging all of this. And now, Dr. Bowen also, I'm sure Dr. Pereira kind of twisted your arm in this. Columbia has been great for taking part and helping us. You guys have really been impactful and really explaining all these things and the effects of the vaccines. I can't thank you guys enough for obviously coming to my rescue 9.5 years ago and giving me the second chance at life. Dr. Marcus Pereira is the medical director of infectious disease. And I have to use my little cheat sheets here because you guys are such esteemed and well-respected doctors in this profession. So pardon me for having to use my cheat sheet, but the list is endless on your credentials and everything that you guys have done. Honestly, the list goes on and on and on. So sorry for using my cheat sheet. Again, my Cliff Notes. But Dr. Marcus Pereira truly is a lifesaver for so many transplant recipients. He's the medical director of infectious disease who specializes in patients with hard to treat complications in solid organ diseases and really goes above and beyond to help all of us that cannot be easily treated. Patients come from far and wide, and Dr. Pereira really goes above and beyond for all of us and really gives that bedside manner. He gives that special touch, and I can't thank him enough. He's come to my rescue on countless occasions for C. diff, for pneumonia, for COVID. The list is endless. So Dr. Pereira, thank you so much. Dr. David Ho, wow. If I could find, I don't know, a ream of paper to list your credentials, it's too many to list. So he's the professor of microbiology and immunology. He's the director of the Aaron Diamond AIDS Research Center. The research lab really has been involved with HIV research from the very beginning since the AIDS pandemic began. During COVID, Dr. Ho's experience led him to assemble a team of remarkable professors, and he assembled just an incredible team, and that's really what led Columbia to be the head. And because New York City was the lead to really bring people to the forefront and people, and hence the transplant journey to come to these meetings to really seek information and to come to this webinar to find out what was going on and to come to Dr. Pereira to find out if these vaccines were safe, to seek knowledge, to seek resources, to find out where we could lead to, because unfortunately some transplant centers weren't giving us the information we needed. So, Dr. Ho, thank you for coming to us and explaining to us what we can look for with long-term COVID and what we can expect and where we need to go to and where we need to look. Thank you for being this esteemed and well-respected doctor in the field. And we thank you for your devotion and this knowledge that you have and this depth and this dedication that you've given. We really appreciate you and we salute you for that. And Dr. Anthony Bowen, thank you for your time and your dedication in this field. You are an internist and infectious disease doctor at Columbia, and I know you spent time in the labs working with your esteemed colleagues at Columbia, and we look forward to developing this relationship with you moving forward at Columbia and hosting some more webinars with you. And thank you for taking part in this tonight. We look forward to hearing from you also. So with that, I'm going to turn it over to Dr. David Ho to talk about the effects of long-term COVID. And really what caused us to want to do something more tonight was an
You
article.
want to listen?
Sorry. What caused you and Dr. Pereira and us to really want to discuss something further. There was an article that was published in the New York Times back in November, that was interesting. It talked about COVID and how it can raise the risk of heart problems for years. And Dr. Pereira and I and Ira Copperman and I were talking about the risks, obviously, because we know cardiovascular issues can come up with transplant patients and recipients. And we thought this would be something really interesting to talk about. So if you can maybe elaborate on that further and what you've found, that I think that would be a great jumping off point for us.
Hey, thank you very much, Jen, and good to see everyone on this webinar. It's a pleasure to join this evening. As Jen mentioned, I'm an infectious disease physician myself, but I've devoted my career to research on viruses. For some 40 years, I focused on HIV/AIDS mostly. But when the COVID pandemic hit, we turned our effort to address the urgent problem. And so we've been working on SARS-CoV-2, the virus that causes COVID-19. And we worked on how this virus has evolved and how does it escape from our immune responses, and how does it escape from antibodies and drugs. So given that this is a transplant group, I prepare my remarks to talk about first our recent findings on a antibody that I suspect some of you are taking or would be thinking about taking. So let me start by sharing a few slides, and then I think my colleague Tony Bowen will take over. Now, could you see my slides?
Yes, we can.
So this is just a picture depicting how the virus has evolved since the beginning of the pandemic here in January 2020 to the last month of 2024. And as you all know, the virus began with the original strain from Wuhan, and that actually
Dr. Ho, you just...
Could you hear me now?
Yes, we can hear you now.
Yes, I think someone was trying to mute me for some reason. But as you could see here, toward the end of the first year of the pandemic, a variant emerged called the Alpha variant, and then there were appearances of the Beta and Gamma variant. And then for much of 2021, we were dominated by the Delta variant that came out of India. And then very abruptly around Thanksgiving time, the new variant Omicron emerged from South Africa, and that we now call BA1. And it just took off with great transmissibility from person to person. And that evolved into BA2, BA5. All of these are now called Omicron subvariants. And then as you could see here, multiple subsequent variants emerge, with BQ and XBB. I know it's really hard to keep track, even for the experts, of these different variants. But if you see this little yellow signal here, toward the end of 2023, another virus emerged in South Africa, but it didn't do very much because it only represented a minority of the circulating viruses. But with a single mutation, that little yellow virus, which we call BA286, became what is known as JN1. And that just took off and dominated much of 2024. And you could see then JN1 gave rise to KP2, KP3, but those did not gain a great deal of traction until KP311 emerged, and that just took off and became the dominant virus for much of the second half of last year. And then in the last few months of last year, this other form called XEC came out. And these are all progenies of this BA286. Okay, and they're highly related. So given this virus evolution, it has had a tremendous impact on the protective efficacy of our vaccines. So the initial vaccine was made with this particular strain. And by the end of the first year, we already know that that vaccine could protect people from catching symptomatic infection with efficacy up to 95%. So it was highly effective. But by the time the virus evolved to Delta, that efficacy had dropped to about half. And then probably dropped to pretty close to zero by the time it was confronted with Omicron. And then the virus evolved into BA5. And by then, we know that the original vaccine was not very protective. So the FDA and the pharmaceutical companies made new vaccine based on BA5 and the original strain, but that vaccine did not do very well. And a year later, an XBB 1.5 vaccine was made, and that was a pretty good booster. And a few months ago, in the US, the KP2 vaccine came out, and we found that it was also a very good booster. Now, given that this is a transplant group, you obviously know that transplant individuals do not respond robustly to vaccines, and hence many would need protection in other ways. And that's when antibodies administered passively could be protective. And many companies, beginning in 2020, developed either therapeutic or prophylactic monoclonal antibodies. Many were developed in the first year, but as a consequence of this virus evolution, what happens is that by the time we got to Alpha and Delta variants, 3 of the initial antibodies were already knocked out by the virus evolution. And by the time we get to the first Omicron, 2 additional antibodies were knocked out. And as we got to the BA5 and the BQ viruses, 3 more antibodies were knocked out. And the 2 that were highlighted in yellow here were the ones contained in the prophylactic cocktail known as Evusheld, which many of the transplant patients and other immunocompromised individuals depended on. And basically for a period of time, there was no monoclonal antibodies that could be administered to immunocompromised individuals for passive protection against COVID-19. But in March of last year, 2024, when JN1 was dominant, a company called Invivyd came out with pemivibart, which was shown to be effective against JN1. But what I think I was tasked to talk about this evening by Marcus was that we had recently found that even that antibody, this antibody that was only authorized approximately 9 months ago, is now much less potent against the dominant viruses today, KP.3.11 and XEC. And we published these findings in a paper that appear in the New England Journal of Medicine. This is a figure from the paper, but I only want to highlight the important part, which is against KP.3.11, you see the activity. Here, it means the concentration required to kill 50% of the virus is 4 microgram per ml as compared to JN1, which only required 0.16. So it's a 25-fold, in our hands, a 25-fold decrease in activity. And these experiments have been performed multiple times, and an average number is about 3.5. So somewhere between 3 and 4 micrograms per ml. So it's now requiring a high concentration of this antibody to protect against this dominant strain. And then for the other dominant virus, we have in a separate paper shown that against XEC, it also requires a fairly high concentration, albeit at a level lower than KP.3.11. Now, this news was not well received by the company that made the antibody pemivibart. So somehow they made the journal to force us to say this is research grade pemivibart. However, I won't go into this, but I will simply tell you that our data on pemivibart corresponds nicely with their data, the company's data, on many other virus variants. So there's no indication that our antibody is any different from theirs. And overall, the concentration required to kill the virus by 50% for the 2 dominant viruses today, in our hands, these are the numbers. The lower the number, the better. So in this case, we show that the virus is actually quite resistant. But the controversy arises because the company claims that it still has very good activity against the dominant virus, although they haven't reported data on the other dominant form. But I would also note that another academic group had results that are in the same ballpark as our numbers. So we would surmise that the protective efficacy of this pemivibart might be decreased. We're not saying it's no longer useful, but its protection would be expected to be lower. Now, I'll skip the technical slide here and just let Tony Bowen tell you about the analysis that he's done based on the data that we have and also based on the company data to show you what is predicted based on his analysis. So Tony, would you like to take over from this point? And then we could go into discussion about some of the points that Jen raised earlier about other issues pertaining to COVID-19.
Sure. Great. Thank you so much, Dr. Ho. And thank you, Jen, for the invitation to join the meeting today. It's great to be able to be here and speak with everyone. So as Dr. Ho was mentioning, in our lab, we found this increased resistance to some of these more recent variants. But the question we also wanted to ask was, how much does that resistance that we see in the lab, in the test tube, how does that correlate to what we might expect in terms of effectiveness in the clinic when we're actually giving this antibody to patients? And to answer that question, I'll just share the slides here again. To answer that question, we looked at some data from previous studies of other antibodies. Is everyone able to see my slides here? Great. So this is just an example of sort of correlation between antibodies that have been studied to prevent COVID and how effective they are at actually preventing the disease COVID-19. So at higher antibody concentrations up in this range, we're seeing effectiveness in the 90 to 100% range. As you get lower concentrations, the antibodies become less effective at preventing COVID. This axis here is also normalized by the different variant. So if you have a variant that's more resistant to your antibody, then that antibody will come down this curve and become less effective. And so we did this analysis looking at both our data from our tests of pemivibart against different viruses and also Invivyd's data. And so looking at the original data that the company developed against the JN1 virus, they saw a very high range of concentration relative to the JN1 variant. And so that would suggest very high effectiveness. And this was part of the way that this antibody was initially approved. And our results against this JN1 variant were quite similar, showing a relatively high range in the concentration, which would correlate to above 80% effectiveness. As Dr. Ho was mentioning, with these other newer, more recent variants, we see sort of increased resistance, and that causes the antibody concentration that we would expect to sort of slide down this curve and become less effective. And so our results are shown here in circles from our lab, and Invivyd's results are shown with triangles. And then these are a series of different viral variants. But essentially, the most important finding here is that the more recent variants, XEC and KP3.1.1, are sort of falling down in this 50% range at 28 days and around 40 to 50% effectiveness range at 90 days. I would also note that Invivyd's data did also see a decline in effectiveness with the KP3 variant, but not as significant as what we saw. So, I think this analysis has to be taken with a grain of salt because really we need clinical trial data, clinical study data to get a definitive answer. But we do see an indication that this antibody is likely going to be losing effectiveness against some of the more recent viruses. But we'll be interested in waiting for some of the clinical study data as that becomes available. So with that, I'll stop sharing my slides and would be happy to turn it over to Dr. Ho again, Dr. Pereira, or any other comments on this.
Perhaps, Marcus, you want to add some comments at this point?
Yeah, absolutely. Thank you, Dr. Ho and Dr. Bowen. The two of you, and Dr. Ho's lab has been a guiding light for many of us as we have gone through the last 4 years, 5 years now, in terms of trying to figure out what to recommend and what to give our patients, right? I mean, if just because a product is available, is it really in the patient's best interest to give or not? And sometimes the data are not clear and you get mixed informations. But I think if you always take it from the perspective of a person who is vulnerable and, and weigh in the risks and benefits of everything that we do, you have to use the data that is available to you to make those decisions. So I have to say, every time there is a new product or changes in the virus, Dr. Ho is the person that we go to for our information. And he hasn't been wrong so far. And that's telling a little bit. The way I take all of this is what I said several months ago to many of you when we talked about bimivibart, and some of the preliminary data that was coming out, that it's sort of, you have to be sort of very thoughtful about who may benefit from this. There might be some benefit. We just can't guarantee that it's going to be a tremendous benefit, and certainly probably not to the same level as Evusheld was. And in that context, you have to think about who is at greatest risk, either of catching COVID or developing very severe forms of COVID. And in our minds, at least clinically, those had been lung transplant recipients in terms of developing severe disease, and those who have received some medications like rituximab. And we talked about, if you receive that particular medication, you're very unlikely to respond to any vaccine, and you're basically completely vulnerable. So I think that, with Dr. Ho's data, I think we kind of have stayed in the same sort of level recommendation that we did, throughout, I guess, maybe the second half of 2024. It can't be a wholehearted recommendation, unfortunately, and we wish it was, but it's just not something that, even if you get that product, the confidence that you're fully protected is just not there, right? You're still going to have to go through the same levels of sort of caution in terms of wearing masks, avoiding large crowds, and still trying to get vaccinated and immunoprotected to the degree that you can. So in my mind, it's not a game changer, unfortunately. But those are my thoughts, and sort of hearing what Dr. Bowen and Dr. Ho have just said is just kind of, to me, that seems the same as it was several months ago. I do think we have some questions.
Yeah, in the chat, there's a question about whether the recommended dose would take care of the loss in potency. And I would let Tony answer that, because his analysis takes into account the dose and the so-called pharmacokinetics of the antibody. So Tony, maybe you could
Sure.
make additional comments there.
Yeah, I think to some degree, what matters is how high the dose is relative to how resistant the virus is. And as you increase the dose, the antibody will stay around longer, so you'll have protection for a longer period of time. But based on sort of the analysis that we're seeing at the current dose that is approved, that's where we are sort of expecting to see that efficacy drop substantially. And the best estimate we have, which is based on previous trials, is that it may drop to around 40 to 50% at 90 days or at 3 months. But that's an estimate based on previous trials and not based on an actual clinical trial with this antibody. Those studies are ongoing, so we'll kind of have to see how the clinical trials change with the current landscape of viruses.
Yeah, I think the company would point out that the concentration of the antibody in the blood remains quite high and at a level higher than what's necessary to kill the virus. However, the concentration of the antibody in tissues, let's say in your nasopharyngeal mucosa or in your lungs, we know almost always is at least 10 times and sometimes 100 times lower. So that is a huge difference. And that's where the protection actually occurs, not so much in blood, because this virus doesn't enter the bloodstream directly.
So there's an additional really great question from Nancy, and I haven't thought about this in a while, and it was somehow it escaped my radar, but the AstraZeneca product, right? I guess Evusheld 2.0, but although there were some major differences, um, I, we had been approached to be part of that study, and then Supernova, I think it didn't really conclude in the US. What's happening with that product?
As far as I know, the Evershell has been removed,
Yeah.
or the authorization has been withdrawn.
Right, right. And there was a new investigational product from the same company, AstraZeneca, but I don't think it's really gone anywhere. The new updated monoclonal from them.
Yeah,
They announced that it didn't neutralize the, what was it, flip originally, the flip mutation.
Yeah,
That's and then the FDA certainly hasn't acted on it. But I think it was that flip mutation that they acknowledged that it didn't neutralize.
yeah,
yeah. I would just add that there are a number of antibodies out there that remain very active against all variants. For example, our laboratory has one that will neutralize everything so far. And there are at least two such antibodies in China which could do the same, one of which is in clinical development. Uh, However, with respect to the COVID pandemic, there's now a great deal of complacency on the part of the government, on the part of the pharmaceutical industry, and they feel that there's very little profit to be made. And yet, folks like many of you are in great need of such interventions. So there's very little interest in taking our antibody or some of the other ones and develop it into clinical products. So I think some degree of advocacy from patient groups would be helpful.
absolutely. I think that's a point well taken. And I think there are many actually individuals in this call that have been very advocate for paying attention to immunocompromised patients in terms of research and sort of sponsoring sort of, the continued sort of investigation for these medications. I think that's a point well taken.
Yeah. I think it's helpful moving forward to know, about some of these, I'm going to say medications, at how we can go about doing that so we can become more proactive in getting these medications to these populations like ourselves.
Yeah.
So we can advocate for ourselves. So if we can get advice and guidance on that, I know there are many people on this group that would be willing to do this, to protect ourselves moving forward. So if we can get guidance on this, I'm sure we would have plenty of volunteers that would help advocate.
That'll be amazing.
Yeah.
Yeah.
Because I know we all want to be safe. We don't want to risk, obviously our gifts that we've been given. So the more protection we can have in this situation, the better. So any help, any advice we can get on this, absolutely. I think we would be willing to do that.
Yeah. So Janet is asking about plasma, so highly purified plasma or convalescent plasma against SARS-CoV-2. There was an approval, right, or a license granted. It is a topic that keeps coming up, and certain areas do sort of use it, and they do have an infrastructure that allows them to use it. Unfortunately, that is just not available you know, in many places,
I
because
don't
it does
care.
require sort of an active blood bank that can purify the antibodies. And unfortunately, New York is just not one of those places. So we don't
It's
really
a seminar on
have that as an available tool.
COVID.
It's also very kind of variable, you know, in the sense that one product may not be the same. I know that some of my colleagues did investigations in convalescent plasma back in 2020, and they've just found, you know, highly variable results with that product. So in some people it did work, in others it didn't. And in some ways also convalescent plasma is always behind too, right? You're accumulating antibodies from prior infections, which may or may not be active against sort of current variants. All of that is to say is it's you know, it's not readily available. If it did, I think it would be very interesting because it is cheap, right? It's basically donated plasma, but we just don't have access to that, unfortunately.
Yeah. If I might just respond to that, the organization that received the license is called One Blood, and they're based in Florida. I think they have offices in North Carolina as well. But if you go to their website, oneblood.org/CCP, the order form is there. And so a hospital has to order it, but they are set up now to ship all over the country. So I think that they've really tried to address the logistics.
Yeah, that's amazing.
And maybe Steve Weitzen, can you just drop the link while I'm talking into the chat so people have it? And so they really, and the other thing that One Blood has done and what they really, you know, have focused on is high quality, high titer CCP, so that it can't be older than 6 months, so that it's much more attuned to what variants are circulating. And so I think that you know, we're very far along as compared to where we were, you know, in 2022, the people that have been working on it. So, and then I was just looking for it. I can't seem to put my hands on it right now, but there's an article that was published out of Charité Hospital that did basically their studies found that if you gave CCP with an antiviral or together early in the infection, that was the knockout punch. You know, it was much more effective than going antiviral. Okay, did it work? Did it not work? Now give CCP if you didn't clear the virus. That if you combine those two. And so I just wanted to, you know, make sure that people were aware of that. Should somebody have the misfortune to get COVID, you know, to just, you know, be aware that this is an option for you, and how, you know, you might have to explain to your doctor how to get their hands on this high titer CCP, but it is available. And they can have it across the country in 24 hours.
Okay.
So
That's amazing. That's good to hear.
I
I'll
want
look
to
into
add
it.
that for people who don't clear the virus and have like simmering COVID, Johns Hopkins is using an antiviral combined with CCP, either remdesivir or if people could take Paxlovid, very successfully.
Yeah, I was going to say
According to
that's
Robin
definitely an area
Avery.
that one can use. We do have a couple of patients with very complicated immunosuppressive conditions, not necessarily solid organ transplant recipients, but sort of, you know, congenital immunological issues that even antivirals like remdesivir didn't work. And that might be an area, like you said, that could work. I mean, I have to say, right now, I don't think we have a hospitalized patient with COVID. And what I think the lucky component of all of this is that our antivirals are still working quite adequately, like remdesivir, Paxlovid. And I think that so far, knock on wood, we haven't had a crisis of drug resistance when it comes to SARS-CoV-2, but it's amazing to have options like convalescent plasma, as we can use either in combination or by themselves.
Right. One other thing I wanted to mention, I think that I don't remember if it's the license or if it says this on One Blood's website, but they talk about giving 1 unit. But the doctors at Johns Hopkins that have done this, you know, done this work to move CCP forward, they say start with 2 units, not 1. So
all right, I can talk to them. I'm guessing it's Bill Werbo and Andrew Caraba and some
actually it's David Sullivan. If you don't have his email, I'll send it off to you.
I can talk to my
And
colleague at Hopkins about it. Yeah.
okay,
If
Okay.
they don't have data on the potency of the recent CCP on the latest variants, we'd be happy to help evaluate that. If
Yeah.
okay,
but we only need a drop of that.
Yeah,
okay,
we can definitely
to-
connect you. I think that would be great.
okay.
because it's important in assessing its potency and efficacy
Uh-huh.
to have such
I
data.
agreed. That'd be wonderful.
Yeah. So to answer some of the questions in the chat, so actually New York Presbyterian Columbia still has remdesivir available as an outpatient. What you need to do is contact your physician, and they set up a virtual a visit and set it up. We're able to. It's still an active thing. Not many centers have that, unfortunately, but we still do as an outpatient.
Virtually
Yeah.
no one has that. Do
I
you have to
know.
Yeah.
do
I
you
know.
have to be a patient of Columbia?
Oh,
That's
good question.
it's a big problem if you do, because virtually none of us are. I mean, some of us
Right.
are, but and I'd love to be there, but I'm not a patient.
Good question. I am not sure. That has not come up yet.
I mean,
Find out.
it's interesting, Marcus. I think you do have to be. You can go on the portal, and like you don't have to like connect with your own doctor. You can do it as an emergency virtual visit. You can schedule that on the portal, and they can write the script for you, and
Very
they're
good.
giving it. Last I heard, in their facility in Queens.
Yeah.
But Steve makes a really good point. I think if they could figure out a way to allow people to access and have that emergency visit uh, that are not current patients, it would be a huge benefit to our community.
Yeah. Yeah. We are, of course, Paxlovid, it's a complicated drug, but certainly effective as well. So it is something that you also have available if you want to stay out of the hospital setting, for example. But those are things. So in some ways, as Dr. Ho mentioned, right, the pharmaceutical companies, we haven't seen any new data when it comes to new drugs and new prophylaxis methods when it comes to SARS-CoV-2 in a while, and it certainly is sort of winding down, unfortunately. So it's definitely something that advocacy is going to be very important. Yeah, Jen.
I was going to ask, is the convalescent plasma covered by insurance or Medicare, Medicaid? That's my next question.
It's a blood product, so I don't know, but I'm guessing that it would be. And it's also it's more in line with the cost of a blood product. So $600 instead of this super high number that we know with them Invivids, Pemgarda. So it's more affordable. But that's a question to get the answer to for sure.
Yeah.
I'll ask and
Yeah.
I'll let you know.
Okay. And then my next question is this going to affect, if you're getting, a blood transfusion kind of thing, is this going to affect, if you need, for kidney, is this going to affect your PRA levels, right?
Well, that's what I was going to say. So, when it comes
something
to
to
a
consider
monoclonal
also.
product, right, it's sort of obviously there was that initial concern, but because it's such a purified product, it didn't really, luckily didn't pan out, right, as a risk for, antibody-mediated rejection or any
Right.
other forms of rejection. When it comes to sort of a more mixed product, I'm guessing that the Hopkins folks are investigating that
Yes,
as well.
they have. We asked this question recently.
And I'm hoping that, in some ways, right, like I guess IVIG, when someone gets IVIG as a transplant recipient, it doesn't really have a very strong interaction with
Right.
rejection. So I imagine it might be the same, but it's obviously an unknown.
Yeah.
Right.
Maybe just to segue a little bit when it comes to COVID, and then we can let Dr. Ho and Dr. Bowen go. I want to share my slides just to give you some context of where we are, and before we talk about bird flu, just where we are in terms of COVID, because obviously we're talking a lot about it, and in no way this is to diminish the risk and obviously the concerns for COVID. But if many of you can see, this is New York City data of cases of COVID. I didn't have hospitalization. I didn't have time to do hospitalizations and death, but the data are there. So we did have a little bit of a blip at the end of December and now the first few weeks of January. It seems like in the last week, cases of COVID have come down. So maybe sort of people returning from trips and travel and the holidays, there was sort of a little bit of a surge, but this number overall, this is where it stands in the grand picture of SARS-CoV-2 in the city and probably our region here, New York metropolitan area. This is where we are right now. And this is where it compares with Omicron, as when Dr. Ho explained at the end of 2021, right? BA1 and BA2 kind of took over, and there were very different variants that the vaccines that we had received previously had very little effect. But it seems like, as you can see, right, this is sort of a very different scenario than what it was before. And then when it comes to hospitalizations, it's the same thing. Even though, there are some cases, we're still seeing very low levels of hospitalization overall. So I guess it's sort of reassuring, but on the other hand, clearly important. We never know what turns this virus will take because it is something that it has continuously surprised us, and it's capable of doing more damage for sure. I don't know, Dr. Ho, do you have any concluding remarks, or Dr. Bowen?
No, I think it's great that you have this webinar and inform each other and occasionally, physician and scientists like us could contribute to. So I'm happy to see this, and in the future, if there's any need for our expertise, feel free to ask. We're here to help.
Thank you so much, Dr. Ho, for taking part in this tonight. We really appreciate your insight, your wisdom, and please, this research that you've done for the past 4 years and throughout the HIV and AIDS pandemic, it's so welcome, and we really thank you from the deepest of our hearts for your devotion to this and all the research that you do it.
Before you guys go, can you answer the question for Stan Day in the chat, please?
Yeah, I'm just texting there as well, but maybe
What was that?
if one has COVID right now, what is the best option? I think remdesivir, and if you can get convalescent plasma along with it. But clearly, sort of remdesivir would be the backbone of your treatment, right?
Yeah.
But that comes first and foremost.
Yeah. I'm just wondering if you guys, I know it's really late, but if you could talk for 2 or 3 minutes about long COVID, anything that you've learned or anything on the horizon about that, because that is a real fear that people, that are immunocompromised have, is getting, COVID and then getting long COVID.
Well, I think Marcus is in better position to speak about that for immunocompromised individuals.
Yeah.
But in terms of non-immunocompromised adults, I think given the, initially it was a prevalent problem, but with vaccination, with treatment, the general impression is that the frequency has gone down. We have other colleagues at Columbia who are conducting a cohort study on long COVID. And at some point, I think it would be wise to involve those physicians and scientists to discuss it. We don't focus on long COVID specifically.
Okay.
Yeah.
Thank you.
Jen, it's a great question. I know that Jen was curious about that too, but somebody has their hand up. So is this a question for Dr. Ho or Dr. Bowen?
Question about flu.
Oh, we'll talk about that next.
Okay, thanks.
One second. So I think in terms of long COVID, you're absolutely right. And we have mentioned this before. We do have an investigator in our division. His name is Dr. Lawrence Pereira, who has established a cohort of patients with long COVID, including some immunocompromised individuals. And you know, it's obviously a very complex problem that I know there are some sort of breakthroughs in the last 6 months about sort of identifying what parts of the virus have elicited this sort of almost autoimmune problem, and sort of, you know, leading to some of these prolonged symptoms. So they're sort of getting close to figuring out, but you know, in practical terms, how to then treat someone, I think it's going to be a more complicated next step. But they are establishing cohorts of individuals and figuring out sort of the variety of symptoms and any connections with the current labs that we have that could give us an idea, right? You're getting better, you're getting worse. This is an area to be concerned or not. There is a risk with heart disease. There's a risk with stroke. There's a risk with depression, for example. So there's all sorts of issues related to long COVID that are still being sort of discovered. So that's going to be an interesting problem. We can bring Dr. Perper and have him give a brief talk. We did do a preliminary study, and other centers have done preliminary studies at a very basic level because it's even very difficult to define, right, what is long COVID. And we did find that transplant recipients and immunocompromised patients did have higher rates of long COVID, at least would meet some of the basic criteria in terms of fatigue, poor sleep, poor concentration, depression, maybe some shortness of breath. But then it gets really clouded because just like the mortality related to COVID, you know, transplant recipients have other comorbidities. So is it because they're transplant recipients, or is it because they already had the other comorbidities like kidney disease, diabetes, heart disease, lung disease, and so forth? Or are those other things sort of driving that issue? I think that yet remains to be teased out. But I would love to hear his thoughts, and maybe we can bring him
Yeah.
next time for all of this.
How do you spell his last name?
Purpura, like the disease. Purpura.
Okay, thanks.
Yeah. Yeah.
I think a question is also like, what constitutes the time duration of long COVID? Is it 3 months? Is it 6? Is it a year? What is really the true definition of long COVID symptoms or the
Yeah.
terminology, right?
I think it's 3 months,
3
but
months. Okay.
some people may be a little bit more strict and say 6 months. I think,
Okay.
but right now, I think it's 3 months.
Okay. Okay.
Yeah.
That's helpful.
Okay. So why don't we wish Dr. Ho and Dr. Bowen good night? Thank
Good
you
night.
both.
Thank you so much.
Thank you.
Yeah.
Thank you guys both very
Before
much. We really appreciate it.
Before I say goodnight, I'll just tell you that for the last 6 months, we have turned our attention to H5N1 influenza, doing the same type of work we have done for COVID.
Oh.
So we'll update you on that, Marcus, at some point.
Thank you. Oh, I'm glad you are. I think I'm about to scare the heck out of this group. So
Well, I think we should be ringing the alarm bell much more than the U.S. government.
yeah, that's exactly right. Okay, have a good night to you both.
Good
And let me
night.
start sharing
night.
Thank you.
these slides
Thank you
on bird
both.
flu.
Yeah, we would love to have you both back, Dr. Bowen, Dr. Ho, anytime.
Okay.
Thank you.
Okay, so I guess let's switch gears right to bird flu because this is definitely has been in my mind for the last 6 months. I don't mean to panic anyone. I think knowledge is power. We need to be aware. You know, there is a lot on the news. And I think some of us tend to tune out because we don't want to hear there's so much else going on. But this is an important topic. And I think it comes from the fact that I don't ever want to be surprised the way I was by COVID ever again. You know, so every time that there is something like this that is, you know, goes into the news, you know, I don't ever want to dismiss it and not pay attention to it. Obviously, I don't want to be panicking all the time, but it really is important to pay attention to what's out there. So bird flu is definitely an important element. So just a big basic about influenza, the virus itself. The things to know about it is that it's got sort of this sort of DNA material, and it comes in 8 segments. Okay, this is how its DNA is arranged, and this will become important because the way it mutates is related to these 8 segments. Hemagglutinin and neuraminidase are important. Hemagglutinin is what binds to the cell when it first infects, and neuraminidase is important because it's what allows the virus to release the new virus to be released out of the cell to infect other cells. The reason why we pay attention to this, because this is a major marker of immunity, and the virus, what specific sort of animals and species it can infect, right? Bird flu, pig flu, or swine flu, for example, or human influenza, and so forth. It has a lot to do with this. And this, as you know, oseltamivir or Tamiflu is active against. So those are sort of some important elements about this. This is sort of a prettier picture from the CDC. And the thing to know about this busy figure is that the hemagglutinin protein, remember I said it has to deal with the binding of the virus to the surface of a cell. This protein of the virus binds to this element on the surface of human cells or in other animal cells called sialic acid. Okay, so this sialic acid is sort of the lock that the hemagglutinin as the key kind of binds to, and then allows the virus to enter the cell and infect and cause all the bad things that it does. Okay, and sialic acid is very specific to the species, right? So human sialic acid is different from cow sialic acid and other sort of animals. But there's a big overlap, and that overlap is what sometimes is an important element of the virus jumping from, chickens to cows to humans and so forth. It's sort of like the ability of this hemagglutinin to bind to sialic acid. All the action is related to that. Okay. Now, you probably have heard this. We all learned this in medical school as two major important elements of influenza is sort of its ability to mutate, right? And it's the reason why, influenza, for example, is sort of something that has persisted over centuries. And now SARS-CoV-2 is sort of this problem that won't go away because viruses mutate, and they mutate in different ways. The most common way that a virus mutates is this thing called antigenic drift, which is just sort of this normal low-level slight change in the virus from generation to generation. And these are just very sort of small little changes. And I think you can appreciate that with that big timeline that Dr. Ho presented with SARS-CoV-2, how the virus over a few months can mutate and sort of, it's very similar to the prior one, but it has slightly differences and then so forth, right? This is called antigenic drift, and it's basically a very common problem for RNA viruses, or feature, I should say. Now, antigenic shift is very unique to influenza A, and it's the reason why influenza A is able to cause pandemics and has caused a number of pandemics in the last several centuries, right? And more recently, the Spanish flu was sort of directly as a result of antigenic shift. And what we might be seeing with this current bird flu is an antigenic shift. So these are features because those 8 genome segments, they can sort of move around between different species-specific influenza. And then if you combine, for example, pig flu with human flu, you get a whole different virus that is able to potentially infect both species or, for example, humans. And if it infects humans and it's a different virus, we're all unprepared and there's no or very little immunity. And that's when we get into trouble when it comes to these pandemics. And it's sort of this reassortment that leads to a new virus, and it's the antigenic shift. Okay, that's the sort of the stuff that really it makes virologists very scared. Now, bird flu, you're going to see different terms. Influenza A H5N1, it's the current one that we're worried about. The other term that you're going to see is highly pathogenic avian influenza, or HPAI. It's a few decades old now. It was first identified in China in 1996. And quite honestly, between 2003 and up until August of this year, people have been tracking it. It really wasn't causing much. There was about 907 cases of confirmed influenza in humans, and it led to 464 deaths. Now, not a lot of cases, thankfully, and usually these were all people who worked directly with chickens or like in poultry markets, for example, and they just somehow got infected from the chicken. The thing that was always really concerning is that, half of those people who got infected died. So obviously that's a terrible ratio, right? 50% mortality. This particular variant of H5N1, this clade 2344B, has become the dominant strain in the last year, for reasons that are unclear, but it probably has high affinity for wild bird populations. And I think you probably saw that in the news, right? Sort of birds all over the world, kind of, whole flocks sort of catching this virus and dying off. Massive, massive die-offs in bird populations based on this particular clade. Now, the thing that really got everybody worried about is that it started infecting different mammals all over the world, whether it was cows or seals or goats, skunks. Now the list is very long, bears, cats. It can infect a very high number of animals and actually have some transmission between them. Pigs were now impacted as of October of this last year. This had been a little bit of a saving grace because pigs are usually the step before getting the virus sort of jumping to humans. So, and the reason why this is concerning, right? Like, we are much closer to mammals in our biology, in particular in our sialic acid composition, than we are to chickens. So if the virus is now effectively infecting other mammals, it means it's that closer to infecting humans, right? And obviously it has infected humans, right? Sorry, this is some data on transmission in cattle. So all over the country, right, cows have had H5N1 infections, and it really kind of impacted their milk productivity. But really, there's a lot of concentration of the virus in milk. And you probably have all read about it, right? So raw milk is a real no-no to be drinking these days because it is full of H5N1. But the good news is that, the pasteurization method that is pretty standard for any commercial, most commercial milk products, kills off the virus. So that component has been fine. I certainly have been drinking milk, and I think many of you have, and we're not catching H5N1. The issue is that people who are drinking raw milk, that they are directly being exposed to the virus. And in fact, actually, there are a couple of reports of cats drinking raw milk and dying from H5N1 at this point. So one has to be very careful with this. These are sort of the outbreaks in cattle by state in the United States, and you can see sort of California as a big dairy product state, has really had a lot of these infections. And in fact, it was the reason why they declared a state of emergency a few weeks ago, is because of all their cattle that are infected. This is just to say that, the cow sialic acid, or the type of flu that is infecting cows, doesn't bind very well to the human sialic acid. So that was sort of a reassuring component, but this is now a little bit old. I think this was published in the summer. So it's not clear that this is still the case, but sort of people are looking very closely, right, at the sialic acid and how the virus can jump. The thing that was a little bit more concerning recently is that now there are reports of pigs getting H5N1, and pigs have been considered traditionally sort of the mixing vessel for avian and human viruses. If pigs can have both of those viruses, then that's when that genetic recomposition of the virus can take place very easily and then start spilling over into humans. And it's all about the sialic acid once more. So, for example, swine flu in 2009, I don't know how many of you remember, but there was a pandemic in 2009 of this like pig flu that actually took over North America. And it was sort of a combination of pigs and human influenza that sort of took over and generated a new virus. The good news about that pandemic is that actually the virus was less severe than the typical human virus. So we didn't really see much. And that was actually kind of a nice sort of lucky aspect about that one situation, right? It's obviously not guaranteed, and certainly hasn't been in the past that the virus, when it mutates, it's less severe. But that was sort of a nice feature at that time. I think many of you know, right? Like, how do you catch this? It's really exposing yourself to a sick animal. So, for example, I have a friend here in Westchester who has chickens in their backyard. I think that's a high risk thing to have at this point, to sort of be in touch with poultry. In particular, if you find a dead bird or sort of, a dead animal, like this is obviously that's sort of common sense, but don't go poking a sick animal or a dead animal. And in particular these days, in terms of poultry, do not really come in contact with them. Obviously, then the real concerning aspect of this was always that we started seeing earlier in 2004 human cases of H5N1, right? It sort of started as a trickle in sort of early summer among poultry workers getting intense contact with these infected chickens, and then coming down with the flu. The good news for the vast majority of these cases that these were also very mild. In fact, actually, most of them were just pink eye, interestingly, that they had conjunctivitis. They received Tamiflu and they did fine. And as of January 25, at least what's confirmed, right? I mean, I think there's a suspicion there have been many, many, many more cases, but there have only been 66 confirmed cases. Now, this is what Dr. Ho was talking about in terms of the government and not sort of putting a lot of weight and resources into this. A lot of this testing has been voluntary, hasn't been mandatory. So, you can imagine a farm worker who their livelihoods depends on working every day for a paycheck. They can't afford having sick leave. So they, one, don't want to report that they're sick. And certainly they don't want to report that they're sick with bird flu because now they're going to be on quarantine for a long time and have no paycheck to sustain their families, right? So there are really contradictions there in terms of all of this testing. And that's why everybody thinks that the number is much, much greater of people who have had H5N1 in the last year than 66. And then, what emerged were some more worrisome signs, right? Like, so far, all of those cases were people who were in direct contact with animals. It was clear where they got it, and they didn't spread it to other people in their households. They didn't spread it to their friends or their families. They got sick and it ended there. So the virus wasn't sort of mutating enough to start human to human spread, right? Which is what we're going to be mostly concerned about. Although there have been some cases in the fall where it wasn't clear that the person who was diagnosed with bird flu actually had an animal exposure. So you can imagine people asking, well, where did you get this? Was it from another person, right? So that's sort of a little bit of a concerning sign. And there have been additional cases. There was one in Missouri, and there's another one in another state. And then the last thing that is sort of, again, you have to put this all together, right? So what if, bird flu jumps into humans and starts spreading between people? If it's not severe, all right, we all get pink eye, whatever, is it really cause for a lot of concern? But knowing what we know about influenza, it can really cause severe disease, right? Many of you have had the flu. I mean, we certainly admit a lot of people with influenza, and some people end up dying of flu. And now we've had a few cases of bird flu that have had severe consequences. There was that patient in Louisiana. It was a gentleman who had a bird flock in his backyard and was exposed, got bird flu. He had a number of comorbidities and was over 65 and ended up dying of bird flu. So that was the first fatality in the United States. There was actually a teenager in Canada who spent several weeks in the ICU intubated because of bird flu. She did fine. She's now discharged and apparently doing well, but and probably was aided because she's young and didn't have additional comorbidities. So that might be sort of a big difference, right, between who lives and who dies, at least in certain forms of the virus. We can skip this. This is not necessarily that important. Things to know about what we have. So the good news is that all the antivirals that we have for influenza, like Tamiflu, for example, they're all active against all the bird flu that has been identified right now. So that's at least some good news, right? It's not going to be like COVID where we didn't even have any treatments available, Tamiflu and the other antivirals are all active against it. So we will have a way to treat this, even if it starts spreading from person to person. Is it guaranteed that everybody's going to do well? I don't know, right? Normal flu, not everybody does well despite being treated. But at least this is something, that it's sort of not going to be an extreme cause for panic. And then they are working on a vaccine. So they have identified several vaccine candidates that have good activity against bird flu, this particularly H5N1. So I think there's been a lot of talk about really producing this massively in preparation for what may come. And this is what I think what you probably would have Dr. Ho look at, right? Sort of, you inoculate a couple of healthy volunteers, you get their antibodies, and then he can do some tests comparing the bird flu and the antibodies and look at neutralization, sort of do the same thing that he's doing for SARS-CoV-2 with influenza. And I think that he's going to be doing a lot of that. So what can we do, right? I mean, and besides, I don't want anybody to panic here, but I think the things about bird flu is we have to really keep a close eye on what's happening. You have to pay attention to this. I think particularly immunocompromised individuals may be at a higher risk, right? Like it is with many other respiratory viral infections. So we do have to be very watchful and careful about this. Clearly, you have to avoid contact with wild animals, farm birds, and other things. Please do not drink raw milk. Please make sure that eggs and poultry are well cooked. I don't think you want to be really doing undercooked eggs or poultry. And obviously, many of you wouldn't be doing this anyways because you are transplant recipients, but it's really an important element here. And you want to get vaccinated for the flu, just the regular flu vaccine that is out there. And the reasons might be sort of a little bit different than what you think. The current flu vaccine that we all got this fall is not active against bird flu. That's not the reason to do that. It's really more against sort of not giving the bird flu virus an opportunity to infect someone who is also infected with the regular flu, the human flu, that then they can sort of exchange their genetic material, and then you have sort of bigger problems, right? So this is sort of something that they're trying to vaccinate all the farm workers and both for cattle, pigs, and poultry, so that they don't are the person who's going to sort of be the one where like both viruses are there and it mutates, and then we have a bigger problem. So those are the reasons. And this is just a last thing, just where we are with influenza. The red line is our current situation here. So it is on the uptick, the regular influenza, both among cases and hospitalized patients. So that's just another great reason to get vaccinated for influenza. And I think, yeah, these are the other sort of COVID slides that we had. I'll stop sharing, and I imagine that many of you will have some questions. Okay, so Sam had a question about xenotransplantation. Yeah, I hope so too. I the nice thing about those pigs is they really are in extremely sterile situations, right? So those xenotransplantation pigs are sort of it's a different sort of situation than farmed pigs. So I don't think that they will necessarily be at risk for this, but who knows if it's sort of widespread. Should we keep Tamiflu at home at this point? I don't think so. I mean, I think that we certainly don't want to sort of have a run, for the pharmacy and like deplete the supplies of this. Obviously, there is a stockpile of oseltamivir available in the United States. So I think that, we are currently, at a sort of the current threat is low, right? This is not spreading among humans right now. I think it's sort of cause for concern, but not panic. And I think we need to pay attention. But as far as like start stockpiling Tamiflu, I don't think we're there yet. So dogs and cats can get bird flu. But having a dog shouldn't increase. I mean, I don't know. Don't have them go around, chasing birds in the park or were sort of like, sniffing bird poop and things like that. I mean, I have a puppy too. I haven't sort of, that's a good question. I have to think a little bit more about that, but I don't think that it's going to increase the risk more than what it is out there. I mean, I think the risk is really going to be when it starts spreading from human to human, right? It's not going to be like dogs spreading to humans. That's going to be the big problem here.
I ask because I'm about to make a commitment to get another one. My dog just died.
Okay.
A very topical question for me.
Yeah.
I do it.
No,
I've
I
been,
have a puppy myself. I know. I need to think about that.
you have to think about it or no?
I have to think about it, but my sense is no. I don't think it's going to increase the risk of bird flu. I mean, I think if the bird flu is really going to come from, these farms and starts sort of infecting them, and then there will be enough of a mutation in some individual that then it's going to spread from human to human.
Thank you, Dr. Pereira.
Okay, someone have their hands up.
Yeah, I was going to say, I see Lisa has her hand up. Dr.
Yeah.
Pereira.
Thank you, because I had a flu question.
Sure. Yeah.
Two items. So A, I was vaccinated for the flu, and B, last week I got the flu. So I was wondering how effective this vaccine round was on flu A and flu B, because I already got A and I don't want to get B if it comes around. Do we have any data
Yeah.
on?
Oh my goodness. So let me see. It's probably a little too early to know the effectiveness here in the Northern Hemisphere, right? Because as you can see, like it's just starting. And obviously, there are breakthroughs. But they do these studies. The way that they sort of look at, or at least predict the efficacy of these vaccines is by looking what happened in the Southern Hemisphere during the months of July, August, and September, which is their winter, right? So I was about to search this. Southern Hemisphere influenza vaccine. And I know I did a report on this last year. So I think it had been okay. I think it had been effective in reducing severe, right? The other thing that's important to understand is, the vaccine, even if there is a breakthrough, it's attenuating the severity, right? And that has been sort of also the message with the COVID vaccines, right? You may still get it, but you're not going to end up in a hospital from it. So I hope you didn't end up in the hospital.
I ended up in the hospital with 100. So that is like my first question is how valid was it? Secondly, kind of cautionary tale. I wasn't feeling well. I called my program. I'm not with Columbia. I'm with another program. And they said, get a home test. The home test said negative. And so I stayed at home for another day until I had to go to the hospital because the fever got too high. And then I got diagnosed and I got the hell out of the emergency room because there's really sick people there and I don't want to get double sick. So I came home and I recovered and I'm great. I got a sniffly nose now. So watch the home test. They're really not that valid. And I actually talked to the manufacturer of the home test yesterday and submitted a report, and it's been documented that it didn't work. My second or third question, I think I'm on right now, is what are you doing with Tamiflu with the duration of the dose for somebody who does get flu? Are you doing the traditional or are you doing a double?
For most people, we do the traditional one, just the 5 days. I think that if someone does require hospitalization, is on the more severe sort of spectrum, then we do have a consideration for 10 days of treatment. And particularly immunocompromised individual who, will just sort of need an extra boost on that level. May I ask which vaccine you got for flu?
I got the big one. I don't remember. I don't have my card here.
Dose?
The high dose, yeah.
Okay. Oh, all right.
Yeah.
It's deflating, right?
I'm
Hopefully
like, why
yours
the hell
is
did
an
I go through
isolated
that?
case, it is sort of, I'm glad you're feeling better,
Feeling good now. And I was just curious if you would go, because I'm on my 6th day, and gosh, that has an awful side effect of a terrible taste in your mouth. And if I could cut it short, but I think I'll just ride it out till Sunday and just keep tasting everything like bleh.
Yeah. probably better off. Yeah. Um, no, I think if you cook chicken and pork and beef and eggs thoroughly, it does really, the virus, it's interesting. Obviously, it's a very powerful virus, but when it comes to sort of heat, it really does sort of, gets destroyed very quickly. And it's obviously the basis for pasteurization, right? You sort of, you overheat the milk and it really destroys all the pathogens. It removes some of the proteins from it too, right? So that's always the debate about raw milk, right? Like you're decreasing its nutrient value by pasteurizing. But I mean, I'll take that versus getting sort of infected from raw milk.
Yeah, I'm not buying into the raw milk thing. Thank you, Kennedy, but no.
Yeah, no.
Yeah.
It's really attenuating, right? I mean, I think that these vaccines, what they do, I mean, obviously, if you can prevent, great. That's sort of the ultimate goal. But people have come to terms that not every vaccine is going to do that. But it really, if it can attenuate, if it can make you survive your infection, that's probably a successful outcome as well. I mean, think about if you compare 2020, right, with subsequent COVID phases, right? I mean, people were dying less and less. Obviously, there's some innate immunity, but even that vaccine was helping to a certain extent, right? So I think that's the idea behind vaccines, RSV, influenza, COVID as well. Yeah, exactly. Exactly. That's the idea. You really either staying out of the hospital or just basically not dying from it.
My question is this, do you think some of this will ever improve? I mean, or does bird flu really stem from all these birds being packed into these tight cages and the conditions that they're surviving under? So they're really just kind of giving it to themselves.
That's a larger question about where we are in this industrial society. Yeah, and that's, it's part of my talk to the medical students about pandemics and epidemics, right? If you think about it, I mean, it is, right? I mean, we are potentiating all of this, right? By having billions of these animals in sort of a setting like that. I mean, it's not the origin of it. I mean, it's sort of these viruses mutate normally. They're always there. They have always been. They will always be there. But we're giving them an opportunity to sort of get turbocharged, right? To spread from chicken to chicken, cow to cow in these sort of very tight compartments. And yeah, we're giving them every opportunity to sort of, do more damage.
I mean, just festers and festers and festers, right?
Yeah. Yeah. There's a very, there are a lot of articles about that now, how pandemics are really sort of the intersection, right, of what we're doing to the environment, our economy, right, and sort of the risks that we're taking by doing all of this. I mean, it's obviously, it's tough to say, right? I mean, this is what the modern world is based upon, right? But we're certainly putting ourselves under a lot of risk. Somebody asked about COVID vaccine. I meant to say that too. So the latest recommendations for the COVID booster vaccine for immunocompromised patients is you have the option of getting it basically every 2 months, particularly during a high season. So it's a personal decision with your doctor and your concerns about your particular level of immunosuppression and potential exposure, right? But yeah, so the vaccine for immunocompromised patients is sort of every 2 months. We're all fatigued from making vaccine recommendations, but that just popped up in late December from the vaccine sort of committee.
I have a quick question.
Mhm.
So was the H1N1 similar to the current bird flu, the one that we had a while back, the H1N1?
Well, maybe some components are sort of similar. So obviously the H and the N, right? Like the H is for the hemagglutinin part of the virus and N is for the neuraminidase, right? And there's sort of different subtypes of each. So there's the H7, H6, H5, H1, and then there's like I think it's up to 3 or 4. Maybe there's a 5 as well. So these are different combinations of sort of slightly different, molecules. But it's probably there's a big overlap, but there's probably enough difference there that our immune systems are not able to be protected against H5N1.
Okay.
And that's always the thing. And that's the same thing that you see with SARS-CoV-2. The virus may be 99% similar or identical to how it was a month ago or the prior variant, but it's that sort of 1%, 0.5% difference at key areas that is really the sort of most important element here that could cause more significant disease. Does that
And then
make sense? It's really sort of, it's
yeah.
about sort of those key mutations.
That makes sense. And one last question. 3 years post kidney transplant, and my labs are fine. That just so thankful. And I've had COVID twice, and survived and did okay without any kind of antivirals and all. But very thankful for that. But my question is that my donated kidney is actually it had the BK virus,
Mm.
and the levels kind of hang around in my urine. They never really go away. They sometimes disappear, but they kind of are hanging in there. And I wondered about the effect of vaccines when you already have this virus on board. And I just wondered if there was any, I don't know, challenge to the immune system.
I don't know about BK in particular. I haven't seen anything about sort of a reaction from vaccines and having elevated levels of BK. BK is a complex virus. That can be a whole hour of talk, and we can maybe bring a kidney doctor to talk about BK. Uh, I'll tell you, I mean, from COVID, from the mRNA vaccines in COVID, we saw a lot of people develop shingles as sort of a side effect of the vaccine. It was mild, it went away quickly. But yeah, there's something about sort of the inflammation that can sort of cause certain viruses to reactivate.
Thank
It's a
you.
side effect, right?
Yeah, thank you. Mhm.
Yeah.
Okay.
Okay, so we have a couple future topics here. BK, long COVID. I think those would be good for the future.
Yeah,
Yeah.
great.
All right. I do think I have to go because I have to put my kids to bed. They're probably like running around over there.
Thank you so much.
Well, thank you. I'm glad we got Dr. Ho here. He is an amazing scientist. So I'm glad I got him.
No, thank you so much, Dr. Pereira. As always, you bring such information to this group, and we so appreciate you showing up and
Thank
spreading
you.
some light, scaring us a little bit on bird flu, but it's great to be
Yeah.
informed on all
Let's
of it.
not panic, but pay attention, okay?
Yeah.
Sam.
No raw milk.
Yeah.
Obrigado, doctor.
De nada.
Thank you.
Thank you
Have a good
so
night,
much,
everyone.
as always,
Bye-bye.
Dr. Pereira.
Night.
We look
Very
forward to our
much.
next meeting with you.
Very good.
Marcus.
All right, take care.
Thanks so much, everyone.
Bye,
We'll see you once again.
Thanks, Jen.
Thanks, guys. Appreciate you all. Take
Thanks.
care, guys.
Good night, Jen. Go eat dinner.
Thanks, guys. I will.
It's actually time for me